Hello, class. Welcome to the next module. So, we just went over genetic inheritance and how to use a Punnett square. Now we're going to do a pivot. We're going to start talking about bio or DNA technologies, different technology that it uses DNA and see their application. For this, there's three things I want to focus on. What the DNA technology is, its function, how it works, very briefly, and its applications. Why is it useful? So for each one, know the name of it, its function, and how is it useful. So overall, though, DNA technology is changing the world. Scientists can manipulate DNA, they can copy it, sequence it, switch it on and off, probe pieces of it, and transfer it between species. I'm gonna check something with my recorder real quick, y'all. So, with that being said, DNA technology is incredibly useful. We've talked about DNA a lot in this class. We've talked about DNA replication, we've talked about transcription translation, or how proteins are made. We talked about inheritance. So we've seen how DNA is used in a lot of functions within biology. Thus, DNA technology is widely important in a wide variety of ways. And today we're going to see that. DNA technology can manipulate, DNA technology manipulates DNA. Manipulating DNA for practical purposes is called DNA technology. I'm gonna highlight that. Skin manipulating DNA for practical purposes is called DNA technology. Applications to DNA technology include learning about life's history, revealing human suffering, protecting against the environment, and enforcing the law. So DNA can be used to learn about life's history, learn about evolution. It can be used to relieve human suffering, treat diseases, protect the environment, see how different toxins, different things that impact the environment, and overall for law enforcement, or getting DNA samples at crime scenes. DNA technology makes use of DNA, as the name suggests. This chapter describes some of the techniques and promise of DNA technology. DNA technology tools include recombinant DNA sequencing, polymerase chain reaction, or PCR, DNA profiling, excuse me, using DNA technology to copy cells and organisms. Stem, this is an example of stem cells research and cloning. And lastly, using DNA technology in medicine. DNA probes and implantation genetic disorders, diagnosis, excuse me, and genetic testing, gene therapy. So DNA technology can be used in a wide way. It can be tools, it can be used to copy cells and organisms, and it can be used in medicine. So DNA technology has a wide range of ways it can be used. DNA technology can be applied to genes and the genome. Remember, the gene is the specific instructions for a protein, versus the genome is the whole set of DNA. And I can write that down on here. Gene, specific instructions. And genome is all the DNA. So with that said, technology can be applied to the genes or the genomes. The genome of any organism can be manipulated, and we'll see some ways how that is. Changing an organism's genes changes the proteins it produces. So anytime DNA technology is used to change a gene, change any sequence in the DNA, it's changing a protein that is produced by that gene. So DNA technology, keep, if we take a step back, remember when we talk about protein synthesis, we compare DNA to a cookbook. The genome is the whole cookbook with all the different recipes. A gene is the one individual recipe, say for like a cake. DNA technology is like being able to go into that recipe, edit the recipe a little bit, change it up to produce a different type of cake. So say if you have a white cake in the beginning, you use DNA technology, you edit that recipe, and then you get chocolate cake. And that's one way DNA technology can work. It can go into the gene, it can change the DNA sequence of the gene, and produce a new protein. So this is when we're going to start getting examples of DNA technology. So DNA technology can be used in a wide variety of ways. For each type of way we talk about, no, its name, its function, like basically how it works, and how it's used. So the first thing, DNA can be expressed in all organisms. In this scenario, sea anemones, excuse me, have genes that encode for glowing proteins. DNA from a sea anemone was introduced to the genome of this fish that we see right here. Oh, let me get my blue. This fish is expressing a glowing protein. So we're taking the gene that encodes for a glowing protein. We are then putting it into the genome of the fish. So we can take a gene from one organism, pull it out, and put it into a new organism. This creates what's called a transgenic organism. A transgenic organism. Its genome contains recombinant DNA. Recombinant DNA is genetic material from another source. So I don't like this definition of transgenic organism. I'm going to give you another one. A transgenic organism is an organism with DNA of another organism added to its genome. And that DNA is called recombinant DNA. So a transgenic organism is an organism with the DNA of another organism being added to its genome. So in this example, we're taking the DNA from the sea anemone that codes for glowing protein. That is the recombinant DNA, the new piece of DNA. And we're adding it to the fish. So the fish then becomes the transgenic organism because it has DNA from a different organism within it. Transgenic organisms contain DNA from multiple sources. Transgenic organisms may be useful, have many useful applications, such as manufacturing pharmaceuticals, engineering hardy crops, and testing human genetic diseases in mice. So, transgenic organisms are used in a wide variety of ways. We can produce pharmaceuticals with them. There are some, especially bacteria, that are now transgenic organisms that can produce some of the medicine we use. Insulin is a big example. There's some bacteria that were genetically modified. They were, they were given recombinant DNA that had the gene for insulin. So the gene for insulin was put in the bacteria, making them transgenic organisms, and thus they produce insulin now that is used by pharmaceutical companies. Crops are also genetically modified. Some crops have genes that are added into their genome to make them more prone to drought, prone to pesticide, things like that. And lastly, testing human genetic diseases in mice. And in research, especially medicine, sometimes we take a gene that we know causes a disease and put it in an organism like a mouse. That we can study the disease more ethically, that we can look at how it functions, what it's doing, and make treatments for it, make medicine for it. So all these are examples of transgenic organisms because we're taking a gene that they comes from some other organism and putting them in it. There's a lot of different ways that these organisms, this method can be used. So remember, a transgenic organism is an organism with DNA from another organism. That's the DNA is called recombinant DNA. It's useful in manufacturing pharmaceuticals, engineering hardy crops, and testing human genetic diseases. Now, how do scientists create organisms? there are several ways to do so. There's several ways to introduce recombinant DNA into an organism, depending on which transgenic organism scientists are creating. Some scientists use viruses to create transgenic or animals. Recombinant DNA combines with the chromosome when early embryonic cells are infected by genetically modified viruses. So you get this, say this is our cell here. That's the nucleus. We get this virus here. It has the DNA for the new. Mark that in a different color. Let's mark that red actually. So you have the new DNA in there. Then after that, DNA is introduced to that new embryonic cell. So the embryonic cell, we have its DNA, but then it's a transgenic organism because it has that one new gene, that recombinant DNA. But then when it starts to grow into the organism, all of the cells, I'm doing this very simply, now have that recombinant DNA in it. So recombinant DNA again combines with the chromosomes early when early embryonic cells are infected by genetically modified viruses. When the cells divide, which is here, as the animal develops, they pass along that recombinant DNA to the entire animal cell. So in the example with the fish and the sea anemone glowing protein, that gene for the glowing protein was put into the embryonic cell of the fish. When that fish developed, all the cells in its body had that glowing protein. So that's why that fish now glows, because the gene was put into it during its embryonic stage by a genetically modified virus. So that's one way you can produce strange um, genetic organisms. So now let's do this question. Which is the following an example of a transgenic organism? Pause. Think about it. So remember, transgenic organisms are organisms with recombinant DNA or DNA from another organism. First one, a human with diabetes who receives insulin protein extracted from a pig's pancreas. So yes, we have a human and we have a pig, but we're looking at the insulin protein, not DNA. So that's out. The algae that is produced by a union of sperm and egg. So this is the union of a sperm and egg. That is, we're not getting a new piece of DNA. We're making something, but that's not exactly right. A corn plant with the DNA from Agrobacterium incorporated into its genome. We have a corn plant, and we're taking DNA, which is right, from this bacterium and putting it into its genome. So corn plant is the original organism. The DNA from this agrobacterium is the recombinant DNA, and we're putting it in the genome of the corn plant. So that is correct. But let's look at the last one. A human with leukemia who receives bone marrow from another individual. So someone with leukemia receives bone marrow from another person. Remember, a transgenic organism received DNA. That one's out. So the answer is C. Because the corn plant is receiving DNA, recombinant DNA, from another organism. DNA sequencing. So now we're going to move on to a different type of biotechnology called DNA sequencing. DNA sequencing reveals the order of the bases. Sequencing DNA can be useful for deducing protein sequences and for determining evolutionary relationships among species. Um, let's define DNA sequence in a very brief way. I'm going to use my highlighter to highlight this. DNA sequence is determining the sequence of bases in an organism. genome. So DNA sequence is determining the sequence of the bases in an organism's genome, writing out all the letters. So remember, the bases are A, T, C, and G. So DNA sequencing is just writing what base is in what place. And this is important for two reasons. Oh, two of which. For deducing protein sequences, basically determining the gene for a protein, and determining the evolutionary relationship among species. So those are the two things about that make DNA sequencing useful. Now, we continue to the next example. Next one is going to be an important one too. It's PCR, or polymerase chain reaction. What if an investigator, what if investigators only have a tiny amount of DNA to examine and need to make more of it? Scientists can replicate DNA using polymerase chain reaction. So the purpose of PCR is to replicate or amplify. I did not spell that right whatsoever. Amplify a DNA sample. Copies of DNA, PCR copies DNA over and over again. PCR separates DNA strands, uses enzymes to replicate each strand, and then repeats the process, creating millions of copies in minutes. So in PCR, you can take a very small DNA sample and replicate that DNA over and over again until you get a large amount of it. And that's why PCR is so useful, because you can start with a small amount of DNA and then end up getting a large amount of that same sequence. We're going to see why this is important in a second. But I want to transition to something else first. So, CRISPR. CRISPR is a very new technology. This is something as I went through college and even grad school, it was first being developed. CRISPR, goofing, allows researchers to edit specific genes. So this is the first DNA, first time we're talking about editing a specific gene. We've talked about inserting genes into organisms. We talked about determining the sequence of a gene, the order of the bases in a gene, in the whole genome too. And we talk about amplifying or replicating DNA. But CRISPR is the first DNA technology that can actually edit or change a gene. The Cas enzyme, which is the enzyme used in CRISPR, cuts the double-stranded DNA using a RNA that binds to the complementary DNA. So we have our gene here. Then we have this guide DNA that goes in, finds its gene because it's a complementary base. It has the complementary bases of the gene of interest. CRISPR allows researchers to edit specific genes again. The DNA is either turned off completely or nucleotides are added to the modified the expression of the DNA. So CRISPR turns off a gene, so you need to turn it off, or add bases to it. Your bases are like A, adenine, thymine, guanine, or cytosine, A, T, C, G. So again, CRISPR is the only DNA technology that can go in and edit a specific gene. That's just like how we talked about with the cake example. If we have a gene that makes a specific cake, say a white cake, CRISPR can go in, change the recipe, and make a chocolate cake instead. So CRISPR is incredibly useful. And this is all the ways CRISPR can be used. Genes have been snipped out of mosquitoes' genome, so the mosquitoes no longer transmit malaria. So one example is of CRISPR being useful is it has removed the gene that um, causes malaria in mosquitoes and stops malaria being spreaded by mosquitoes. Plants can be altered to boost yield, improve disease resistance, and survive drought. So CRISPR can go on to edit plant genomes, plant DNA, and crops to make them more robust, make them more resist droughts, make them disease resistant, or even just make them more produce more of their product. Chicken eggs could be engineered to reduce allergies in humans. So CRISPR can be used to remove the protein that someone is allergic to in eggs and make them allergy free. Broken genes that trigger illness in humans can potentially be repaired. So potentially, and this is something that's still in the works, if a gene is malfunctioned in someone, there's the potential CRISPR can go in and edit that gene specifically to make it functional again. And I say that potentially because that is something that's new that's being worked on and it's not yet in practice yet. But if there is the potential, since CRISPR does edit a gene, if we have a gene that doesn't work, CRISPR can go in to fix it. Lastly, cancer cells in mammals could be modified so that they are more effectively targeted and eliminated by the immune system. Saying we can target specific cells using CRISPR, change their genes, and change their function or make them easier to target and remove. So again, CRISPR is widely important because you can go in to change an actual gene. And if you change the gene, you change the protein, which has a lot of beneficial effects down the line. So now we're going to go to DNA profiling. DNA profiling detects genetic differences. Investigators use PCR to amplify DNA from tiny biological samples. Once they have enough DNA, they use DNA sequencing to detect genetic differences between individuals. So again, we talk about PCR. PCR is polymerase chain reaction. It's used to get a tiny amount of DNA, amplify it to boost it to replicate it into a larger amount. And that's important in DNA profiling. We can get a small amount of DNA, we can increase the amount of the DNA we have using PCR, and then we can compare it between individuals. Investigators rely on single nucleotide polymorphisms, SNPs, to compare DNA samples against databases, a database of known samples with SNP combinations associated with known phenotypes. So, SNPs, read my highlighter. Oh, no. And this, I don't want you to know the specifics, but I want you to know the gist of this. And I'm going to break it down for you. SNPs are single nucleotide polymorphisms. Basically, a change in the single nucleotide of the DNA. So here we see in version 1, we have AAGC GTA. By version 2, we have AAG, all the same, A GTA. So that one nucleotide is changed. So in DNA profiling, we can determine that single chain. We also look at short tandem repeats, or STRs. These are short repeated sequences in DNA. And we see two samples here. In the pink, these numbers, 1, 2, 3, 4, and so forth, are the short tandem repeats. And the top one, we have 8 repeats. This sequence repeats 8 times. But in the bottom one, it only repeats 6 times. So DNA sequencing shows how many short tandem repeats, STRs, are in a person's genome. Everybody may have a slightly different amount of these STRs, such as the um, top one, the top DNA sample has 8 and the bottom one has 6. So when we do DNA profiling, we can look at STRs to see if the DNA sample matches someone or not. SDR patterns are useful in criminal justice. Law enforcement agencies have identified a combination of more than a dozen SDR sites in the human genome that can match individuals to blood, hair follicles, and other biological evidence left at crime scenes. So again, by looking at the STRs and different ones, we can determine the likelihood of a DNA sample matching somebody. We can determine who was at the crime scene using that DNA sample. And the first step, again, is PCR, preliminary chain reaction. We get that DNA sample, we amplify, we replicate it to get a lot more of it, and then we compare the STRs to see if we can find the subject that matches that DNA sample. Next is stem cells. Stem cells are undifferentiated cells. Stem cells are undifferentiated cells. Stem cells are special because they can develop into different types of cells. So stem cells are like the wild card of cells. They can change into different types of cells. Maybe we talked about germ cells and um, reproductive cells. Germ cells are normal cells in our body. Reproductive cells, or gametes, are sperm and egg that produce new offspring. Well, stem cells are the cells in our bodies that can change into other types of cells. Scientists are using stem cells to develop treatments for many diseases and to regenerate damaged body organs and tissues. And that's why this is so important. This is why stem cells are so important. We can treat different diseases with them, and we can use to regenerate damaged body organs and tissues. And there's different types of stem cells. Embryonic stem cells that are totipotent. Totipotent, these are embryonic stem cells that give rise to all types of cells, all cell types. So an embryonic stem cell are totipotent. They can produce any type of cell in the body, and it's incredibly useful. So these embryonic cells can be used in a wide variety of ways because they can produce any cell in the body. This is different. If I can let go, I want to give me one second. We're having a little technical difficulties. There we go. Adult stem cells. Adult stem cells are pluripotent. Adult stem cells only differentiate into a limited number of cell types. So embryonic stem cells can make anything. Adult stem cells only make a couple different cell types. Stem cells in the bone marrow, for example, differentiate into all blood cell types. So within the bone, we have this thing called bone marrow. And within the bone marrow, we have some adult stem cells that are used to produce all the cells in our blood, red blood cells, white blood cells, and so forth. So remember that stem cells are cells that are undifferentiated. They do not have a function. They can just change into another type of cell. Embryonic stem cells can change into any type of cell, while adult stem cells can change into only a certain amount of cells. Now, and again, stem cells are very important because they can be used to treat diseases or regenerate damaged body organs or tissues. Almost there. Couple more. Cloning. So cloning is also another DNA technology. Cloning creates the exact copy of an organism. Cloning, or asexual reproduction, is common among single-celled organisms such as bacteria, archaea, and many protista. Plants are very easily cloned as well, though. Remember, Mendel would take pea plants and have them self-pollinate at times. That creates a clone of the plant. So cloning is making an exact copy of an organism. Now, in more complex organisms, we can't just make a copy very easily. We use what's called somatic cell transfer. Somatic cell transfer is used to clone animals. The first cloned animal was Dolly the sheep. The adult sheep's DNA was extracted and put into a different sheep's egg cell and implanted into the surrogate mother sheep. After developing from an embryo, Dolly was born. So what we did, we had Dolly here. We took some of the Dolly cells and took out the nucleus. The nucleus had all the DNA for Dolly. On the other hand, we took an egg from another um, organism, another sheep, and we removed the DNA. We removed nucleus, removed the DNA, and then we combined that DNA and the nucleus together to get a new cell that just has Dolly's DNA. The embryo divided, was put into a mother, another sheep, and we got the clone. And that clone, because it had the DNA of Dolly, was Dolly again. So in complex organisms, we can take the DNA from a cell, put it in an embryo, and create a clone of that individual. And now this sounds like a very simple process, but it's very complicated. It's harder to do, and there's a lot of ethical issues with it as well. But this is how we can do cloning in more complicated organisms, such as sheep. And cloning could be important for a variety of reasons as well. DNA probes are next. DNA probes detect specific sequences in the DNA. So that's their function. DNA probe detects specific sequences. Many medical tests and procedures use DNA technology. For example, DNA probe can be used to test whether a patient is going to inherit a cystic fibrosis allele. So DNA probe is a marker for DNA that's used to see if a sequence is present in the DNA sample. In this case, we're taking, excuse me, the probe, this probe right here, and we want to see if that DNA sequence is in the DNA of the individual. If it binds to that DNA, we know that DNA sequence is in the individual. But if it doesn't bind, we know that that DNA sequence is not present. So probe is detects a specific DNA sequence. DNA probes are single-stranded. A DNA probe is a single-stranded sequence that is complementary to a known region of DNA, such as a cystic fibrosis allele. The probe is labeled with this fluorescent tag, which is this. The researchers can then detect the allele using the wavelength emitted by the probe. So once that probe is attached, like in this example right here, we can look for this fluorescent light that's given off by the probe. And if we see that fluorescent light, we know, hey, that DNA sequence is there. But in this example, the DNA sequence isn't there. So this whole probe is going to get washed away and removed. Thus, we don't get the light anymore. Genetic tests can detect existing diseases. DNA probes can test for many different genetic diseases in embryos, in children, or adults. So again, the probe can be used to test for different genetic diseases in individuals because it looks specifically for the gene that produces that disease. The last thing we're going to look at is gene therapy. Gene therapy replaces faulty genes. Gene therapy may someday provide new treatment options for genetic diseases by replacing faulty genes in a person's cell. So the goal of gene therapy, and this is a growing DNA technology. Gene therapy is something growing, it's still something being produced, it's not widely available just yet. There's a lot of hurdles that need to get over for gene therapy to be more prevalent, more widespread. But someday it can provide new treatments to genetic diseases by going in to replace the faulty gene. Gene therapy uses, and again, it uses viruses. This I'm not going to go over too much. But an example of genetic how gene therapy will work is cystic fibrosis. Um, for example, cystic fibrosis is caused by a poly gene on chromosome 7, the result of the abnormal CFTR proteins and lung cells. Healthy versions of the genes are placed inside viruses which the patient inhales. And once they inhale it, that virus then infects the cells that have that faulty gene and replace the faulty gene with a working one. And then you have a cure for cystic fibrosis. Gene therapy gives cells healthy genes. The virus injected the genetic material, including the healthy gene, into the lung cells. The lung cells now produce a normal protein. So we take a virus, we put the healthy gene in it. We infect that virus into the patient. It injects that DNA that with the healthy gene into the patient. And now the patient has a working protein because of that. And that's how gene therapy works. But again, keep in mind this is a growing tool. It's not widely available yet, it's not widely used yet, because we are still learning more about it and how to make it effective. So that is it for today. Again, know, I'm going to go all the way back. Know the big DNA tools and what they're both how they function and what they're used for. So know DNA probe. I want you to know mm, cloning in general, cloning and specifically the somatic cell nuclear transfer. I want you to know stem cells, both what they are and how they're used, the way they're used, DNA profiling, CRISPR, PCR, DNA sequencing, and transgenic organisms. So know each of those, how they function, and what they're used for. Like always, if you have any questions, reach out to me, let me know. I can always help you out.